Dr. Jesus Antonio López Carvallo. Please welcome. I welcome you, Dr. Jesus. Please have it.
Thank you for your kind introduction. Thank you for HRI committee for the invitation. I'm happy to be here. So today we are going to talk about why we use homeopathy in aquatic organisms and how we think is working homeopathy over the biological system of these aquatic organisms. So we are first start presenting the team. Meet our team. We are the Aquacultural and Agricultural Homeopathy Group. The head lab of this team is Dr. Mason. It has a star here. And I was his student as a master and PhD student. Right now, I'm working in Ensenada, Mexico. Dr. Mason is in La Paz, Mexico. And I'm working with Dr. Cruz, looking at the interaction between pathogens and host, using functional genomics. We are more than 30 researchers, students, and producers. And recently, I'm moving to Chile, so we are going to have many collaborators in many parts of the world. So first, let's start defining aquaculture. It's a pretty simple definition. It's the farming of aquatic organisms. But this farming implies basically the intervention of humans. We can control stocking densities, feeding, protection from predators. So we want to increase the production of aquatic organisms. These farmings need an owner. So this is an economic activity. And the main goals of this economic activity is basically increase organisms growth rate. We want a profit of that. If we can increase the growth rate of these organisms, we can increase our profit in this economic activity. We also want to reduce operational costs, make more efficient the use of nutrients that we use to feed these organisms, and keep the organisms alive. After all, it's an economic activity, so we need something to sell, right? We have a little problem here. The production of aquatic organisms is expected to be increased. The demand of marine organisms is expected to increase due to the projected increase of population around the world. We have two options here. We have aquaculture and we have the wild catch, which are fisheries. Unfortunately for the fisheries, it's an extractive production, so they already get a limit. So if we want to increase the production of marine organisms, we are going to need the use of aquaculture. But in this way, we need to project this production or increasing production of aquaculture in a sustainable way. That's why we are going to talk about homeopathic medicines. Understanding sustainability as something that you can get from the natural resources, you can take a benefit of natural resources, but you have to make sure that those natural resources are going to be able for future generations. We have to care about environmental health, we have to care about community development, and of course, we have to care about profitable because we are working because we want to make money with this activity also. However, it's not that easy. We have some major challenge in aquaculture production in a sustainable way. I divide this in three blocks: health, innocuous, and sustainability. And in the health, in aquaculture, what we use to grow the organisms is using high stocking densities because you can produce more organisms in a reduced area. You increase the profit again. However, when you increase high stocking densities growing these organisms, you increase the stress of the organism, and also you start to reducing the water quality. And this is the perfect mix for pathogenic bacteria to proliferate. In aquaculture, we have different bacteria. Some of them are opportunistic, like vibrios, that are always, or is very common to find them in culture water. And when we have this perfect mix, they increase, and the organisms are more susceptible for disease. some producers, to avoid these negative effects of potential pathogens in the water, use antibiotics and disinfectants. However, these chemicals affect the benefic host microbiota and also promote the selection of antibiotic-resistant bacteria. Also, these chemicals can be accumulated in the tissues of the organism, so it's something that is not desirable. I want to make a highlight when we use these chemicals in a prophylactic way. And also, in aquaculture, we have effluents, which is wastewater from the culture activity. And these effluents mostly came with high organic matter, disinfectants, antibiotics, and can have the presence of multi-resistant bacteria. As a way to make a potential solution to these problematics, we have proposed the use of high-diluted bioactive compounds. We are going to take later why we use this name, high-diluted bioactive compounds, but they also integrate the use of homeopathic medicines. So our definition for this high-diluted substance are basically that are formulated by serial dilution. You are going to be very familiarized with this definition. We mostly use decimal and centesimal potencies, and we have an energetic agitation for initial concentrates. They have diverse origins that can be minerals, plants, cells, pathogenic bacterial lysates. And we believe that most of these substances are acted under the principle of hormesis. We have a biphasic dose, a biphasic response dose, where there's a same compound that may cause a negative effect in a higher dose, it can have a beneficial effect in a lower dose. See? It works under the principle of low doses and the principle of like cures like. How will you, or how we visualize this interaction with oyster larvae? Well, what we know that happened when we challenge oyster larvae with an inactivated pathogen or a killed pathogen or a compound from that pathogen is that that larvae, it has to recognize those pathogen-associated molecular patterns, those PAMs. And this is done by receptors. The larvae has to internalize that signal of danger. So then this larvae can activate this defense mechanism and also an antioxidant response. This activation of the immune response will depend on the dose of the substance that we are being used. So we have here an example of using, let's say, an endotoxin that can be found in pathogenic bacteria, gram-negative bacteria like polysaccharides. It doesn't matter we don't have the bacteria, but we have a component of the bacteria. The larvae can recognize that, that molecule that has a potential danger for his health. So according with the concentration of this molecule, the response is going to be different. When you have a very high load of these lipopolysaccharides, you are going to damage the cells or the response of the larvae is going to be so strong that it's going to affect himself. However, if we reduce the dose, we can have a benefit effect, and we can prepare the oyster larvae to be challenged with other pathogens and increase survival. There is a wide variety of high-diluted bioactive compounds. Each of them will have different interactions with the biological systems of the organism. So, summarizing what we just told, we have first a biocommunication by specific receptors. Somehow the molecule has to be recognized, and then the signal has to be internalized by signal transduction. which is related to modulators and transcriptor factors of the organism. And then this is going to promote the transcription of genes related to that specific receptor. That's how it's supposed to be working the cells of the organisms that are being faced by a pathogen or for something else. So this activation of the biological response is what we study. These interactions are very different. This is from Bellavite, 2014. We have here the traditional communication. There is a molecule that can be recognized by the receptor, but also there is this other hypothesis that there is not a molecule like that, but also can recognize or can interact other things like biophysical communication with these receptors. But what we have clear here is that we need an interaction with the receptors so the cell can internalize that sign and can have a response to that effect. I think there is very interesting this table because it gives us an insight of how much diluted is the original compound. This is just an example using a nanomolar concentration in the mother tincture. So we can see that the lower the potency, you may find some compounds of the original compound. So this may have a different biological effect depending on the potency that you may be using. That's what we are suggesting here. We don't know this for sure. This is according to what we have read in bibliography, where the low dilutions may be related to possible presence of original molecules. And the higher the dilution, you may have, there are some authors that report nanoparticles, but also biophysical communications. And what I have seen in this meeting is I see that this is still very controversial, right? So after the potency 12, that we are beyond the Avogadro's number, is where we have been found also nanoparticles detection. So our efforts to evaluate homeopathic medicines in marine organisms have mainly focused on understanding how these compounds work and which dilutions have better results in the performance of the organism. This action should reduce time and effort when characterizing homeopathic medicines components. The advantage with working with mollusks and crustaceans is that we are avoiding the placebo effect, right? So for me, it's really hard to understand how an oyster can have a placebo effect, right? So the main challenge that we face here using these homeopathic medicines was we are using a very, very diluted compound. We are using a homeopathic medicine. How we make sure, because we work in tanks, that this very, very diluted or this homeopathic medicine is getting to all the organisms we are culturing. For a human, it's easy because you can use a pill, you can just put it in your mouth, and you can make sure that the human is taking the medicine. But how we can make sure that 100 oysters, 1,000 oysters are getting this homeopathic medicine? So we made a prototype. This is intellectual protection from Mason Swastegi, to make sure that the mix is going to be homogenized in the system and it's going to be distributed to all the chambers where we are growing at the organism. In marine organisms, we formulate treatments from Vibrio lysates. We are going to talk mainly with these formulations, Vibrio lysates and silica, but also we have proved other commercial homeopathic medicines that are used for human or veterinary use. We have used Pasival, which is recommended for relaxing properties. The organisms are stressed, so our logic was, well, they are stressed. Let's relax these organisms and let's see how they perform. we use siliciateran silica and phosphoric acidum because they are related to increased food assimilations. So if we want to increase profits, if we want to have a better use of the diets we are giving them and reduce operational cost. And we have the formulation. This formulation is made by our team. It is formulated by 2 pathogenic vibrios. This is a high pathogenic bacteria for aquatic organisms. And this was we think to use this because they cause major challenge and problematics when we grow these organisms at hatchery in laboratories. The way we evaluate these homeopathic medicines is mainly by prophylactic use. We want to prevention the disease because corrective measures are more expensive. So we want to make a less expensive manage of this aquacultural activity. And when we evaluate the prophylactic use of these homeopathic medicines, we also prove if they are working, and we face the organisms, and we have a challenge with pathogenic bacteria, which are mostly vibrios, and then we see the response. We are interested in measuring, which is the growing, the survival, productive parameters, the histology, how the tissues change the arrangement, or what kind of cells are being proliferated by the action of these homeopathic medicines. We also evaluate the biochemistry level and differential expression, the expression of genes. So I want to talk about some things that we have done in the lab. I'm going to present you some works in marine fish, in crustacean shrimps, and in some mollusks. And in general, for all the aquatic organisms that are being cultured in aquaculture, we have this major challenge that the infectious disease they are one of the main limiting factors for marine organisms production. And also, there is a bottleneck where we want to increase the use of nutrients. So they can be linked to increased growth and reduction of operational cost. From now on, I'm going to start to talk about silica and phosphoric acid as seed and Pha, just to talk faster. And for vibrios, I'm going to say Bia and Bip. And I'm going to talk about another homeopathic medicine, which is formulated by endo and ectoparasite. This has been used only in one study, which is this. This is our first study. We evaluate this homeopathic medicine, Passival, Pha and Seed, and Eve. These fish were already with a parasite in the intestines, which is Coccida parasite. And we treat them with homeopathic medicines. And what we saw is basically that when we add these homeopathic treatments, a good thing is that all the organisms are growing the same. We are not having negative effects. And we have a very interesting thing here that is pointed with the red arrows. We have that Passival and Eve that contains or is formulated by ecto and endoparasites. This is an endoparasite, Coccida. We saw an increase in lymphocytes, which are immune cells from the fish, accompanied with a decrease on intestinal Coccida that we can see it here, compared with the control, and an increase of survival, which suggests that the activation of a specific immune system of this fish is being activated or is being benefited by the homeopathic treatment Passival and Eve. On the other hand, we saw that Pha and Seed also had an increase of mucus in gill, and this mucus is mainly related to the production. His production is mainly related to the protection against disease. Also, passival and PAD-seed induce the accumulation of carbohydrate reserves in liver. So I want you to put attention to this. With silica, is increasing energetic reserves in liver in fish, which liver is really a really important organ for carbohydrate storage. We have another experiment using 30 days post-hatching fish. We treat them with beef, bia, and seed and fat, 7 centesimal, and we evaluate the immune response using modulators of the immune response and effectors of immune response. We have an interleukin, a pro-inflammation interleukin also. And these results are very interesting because we are looking at overexpression of immune-related genes with the bia and beef treatments before the challenge. But this doesn't happen with the treatment seed and fat. Only, however, when the fish is challenged against a pathogenic bacteria, even seed and fat didn't increase the expression of these immune-related genes. When they are faced with the pathogen, then increase the response, and this response is much higher than the controls, which are here in gray. This is very interesting because it's telling us that these homeopathic medicines, when they are used prophylactic, they don't always activate the immune response before the challenge, but sometimes they do. Because we saw that interesting result with the increase of carbohydrate reserves in liver and fish before, we decided to analyze the content of or the activity on digestive enzymatic activity in 30 days post-hatching fish, and we didn't saw an effect of survival, which is good. It's not affecting the health of fish. By the contrary, well, in the case of bia and beef, we didn't saw a specific regulation of this digestive enzymatic activity. However, when we use seed and fat, we are looking here an increase of trypsin and lipase, which are enzymes related to protein and lipid assimilation, and a reduction of amylase. And this is something positive for these fish because when they are larvae, they are mostly fed by carbohydrates, so they increase this production of amylase, but they change their diet and they need to increase the activity of trypsin and lipasa because they became carnivorous. This means that we can reduce or we can accelerate the digestive tract maturation with silica and fat of this fish, which means less operational cost for us. So we can conclude with these results that bia and beef had a higher modulation of the immune response, which are formulated by Vibrios. They kind of modulate digestive enzymatic activity, but it's not so clear. However, when we use seed and fat, we have an increases of energetic reserves. We also enhance growth of the organisms, which is how is it used in veterinary. It has a higher modulation of digestive enzymatic activity and enhanced immune response, but only during the challenge, at least in fish, right? Does it make sense? Well, these compounds had been related in some way in low doses to activation of immune response because the interaction of receptors of the cells that are present in fish or other organisms. So the question here is, can high-diluted bioactive compounds or homeopathic medicine induce the same or similar biological response in other marine organisms? And the answer is, yes, they do. So this is really interesting because we are not only replicate the same results in just one organism, but it can be replicated in different organisms. And we are talking about an invertebrate. And now we are going to talk about invertebrates, which are marine crustaceans, which we work with shrimps, and we are working with bivalves. So the first example here, we use the same homeopathic medicines. And here happens something really interesting, because in our research group, we work with different things. We work with probiotics, we work with vaccines, we work with immunostimulants, and we include homeopathic medicines now. And what we are looking here is basically that we have that both treatments of Vibrios reduce the load of Vibrios, which is something that we desire, in water and the pathopancreas of the organisms, even lower than the positive control. Our positive control is a Streptomycetes. This is a probiotic that we use in our lab to inhibit the Vibrio growth. So what our guess here is that we are activating somehow the immune response of these shrimps. So they are acting like destroying these Vibrios, not only inside of them in the pathopancreas, but also they are reducing the load in water. Both treatments increase growth and survival, which is something desirable for production. And additionally, both treatments increase, additionally, in a second experiment, we only assess the effect of SID and FA. And again, as we saw before in fish, we looked at the enzymatic or digestive enzymatic activity is increased compared with the controls when we use SID and FA. So we are looking that this treatment with silica and phosphoric acid is increasing energetic reserves, and it seems related to the increase of enzymatic activity. Now we have another example with Catarrhina scallop. We measure here the immune response, but we did something different approach. We used an even lower potency, so we wanted to be more near to the mother tincture, and we also used the 7th potency, that is the most often used potency that we use in our lab. Also sometimes 30 potency, 30C potency. And what we are looking here is the hemocytes, which are the immune response cells from scallops, quantification, and we are looking enzymatic activity of antioxidant response. What we are looking here is that both treatments formulated by Vibrio increases modulatory response. So it replicates again. When we use it as a prophylactic treatment, the immune response is not highly modulated by silica, but again, by Vibrios it is. Also, we look at an increase of carbohydrate energetic reserve when we use silica and phosphoric acid. So we are looking the same response in different organisms here. I'm going to be a little bit faster. So these homeopathic treatments enhance growth, increase energetic reserves accumulation of marine organisms, and it had been related to higher digestive enzymatic activity, this increase of energetic reserves and growth. However, this treatment modulates immune response increases and survival, and when not treated organisms are challenged against high virulent pathogenic bacteria. However, I wanted to get at this point. However, immune response is not always activated before the challenge, but is during the challenge. So that's what we wanted to explain. So this is the interesting part. We need to understand how these homeopathic medicines work for a better decision making about their use. However, articles and documenting the way of action of these marine organisms is scarce. So we are kind of walking in a black area, and we are trying to put some points to see how these interactions are doing. So I'm going to skip this part. This is a transcriptomic profile where it's telling us that the treatments of Vibrio and silica and fat, they are clustered in different groups, which is something desirable. and they are activating the immune response of the organism. And we look at a higher number of regulated transcript in mantle compared to gill. So we think that these homeopathic medicines, at least in the scallops, are being detected or are being interacted with the mantle tissue of the organism, which is the main tissue exposed to the environment. All the treatments here upregulate genes related to toll-like receptors. And all the treatments activate pyruvate related to internalization and elimination of molecules related to pathogens. However, none of these treatments regulate the main modulators and effectors of the immune response. And yet, when these are treated, the survival when the organisms are challenged against a pathogen, they increase survival. So how can it be that they, yeah? Okay. Yeah? Okay, do I keep? Okay. This is the pretty interesting part because we are looking that sometimes the immune response is not activated, but when they are challenged or faced by a pathogen, the immune response is activated. So how can we explain this? We are looking that the main modulators of the immune response are not being there, but we have a clue. We have an activation of non-self-recognition process. So our guess here with those results is that when we use homeopathic medicines, we are only activated toll-like receptors, which are highly involved in the recognition of pathogens. And this interaction also increases the abundance of genes related to the arrangement and ubiquitination of protein suchamembrane receptors. So we have also this biomarker related to cytoprotection that keeps the homeostatic levels of the organisms or the cells, and we have apoptosis inhibition. Our guess here is that the organisms are detecting something as a potential danger, so they increase the chances to detect that danger because it's in a very maybe a very low dilution that the cells say, hey, something is wrong here. I have to increase my chances by proliferating my toll-like receptors to see if I have a chance to detect what is going on there outside. However, stop, don't kill the cell. The problem, it seems to be there, but we don't have to rush and enter in the apoptotic inhibition. So all these biomarkers have been linked to the recognition and internalization of PAMs. And we can say that organisms mainly activate the non-self-recognition and internalization of PAMs without properly activate the immune response. This may explain why these homeopathic treatments have an advantage to overcome infection disease. So these are our main conclusion. I'm just going to stop here to let some time for questions. Fortunately, I know I couldn't explain everything in very highly detailed, but we published an article. I am proud of this article because it's been pretty hard to publish this kind of works in aquaculture research. I work mainly in aquaculture field, so I try to publish in aquaculture journals, and I've been rejected like many times. But this manuscript was accepted, and it includes all the information that I just talked today. Also the dynamics that we think how the mode of action is being done. Also, we have the ResearchGate profiles. Feel free to contact us, Dr. Masson or me. And thank you very much for your attention.